So the title of my talk is Protein Methyltransferase Inhibitors as Personalized Cancer Therapeutics. So I just wanted to give some disclosures. I have the following financial relationships to disclose. I received grant and research support from LLS, MMRF, GSK, ASI, and Celgene, and I'm a stockholder and an employee of Epizyme. And I will not discuss off-label product use in my presentation, especially because we don't have labels yet. So just to give you a little agenda of what I'm going to talk about, I'm first going to give you some background on histone methyltransferases and their potential therapeutic value in cancer. I'll then transition to tell you about DOT1L in mixed lineage leukemia, and then I'll talk about EZH2 in non-Hodgkin lymphoma. It's kind of a nice follow-up to Lou Stout's talk. And then I'll close out by talking about a role for EZH2 in malignant rhabdoid tumor. So Jonathan Wettstein did a great job of introducing chromatin structure and epigenetics, so it makes my job much easier. But I wanted to reinforce a couple of points. Over the last decade, it's become increasingly clear that the chromatin structure plays a very important role in regulating gene transcription. And additionally, we've begun to think of histones not as just structural proteins, but they are, in fact, important nodes in signal transduction. So we're looking here at a blow-up of a human chromosome, and what you see at this highest magnification is just a cartoon of the classic beads on a string sort of model. And what you have here is about 150 base pairs of DNA wrapped around histones. And these histones, you should keep in mind, are actually octamers. They're made up of two tetramers of histones H2A, H2B, H3, and H4. And what you can also see in this cartoon is, as Jonathan mentioned, is that there are actually two really distinct forms of chromatin in higher eukaryotes. You can see that there's euchromatin, which has this looser compaction of the histones, and it's transcriptionally permissive. The transcriptional machinery can access genes in this context, and gene transcription can be turned on and off. Contrast that with heterochromatin, which is more transcriptionally repressive, much more tighter compaction of the histones, and that sort of blocks access to gene transcription. So one of the important mechanisms for regulating chromatin structure is, in fact, post-translational modifications of histones. And so there are a variety of different enzymes which have activity at the level of the histones, and I'm just giving you a feel for some of those. It can be things you're very familiar with, like phosphorylation, ubiquitination of lysine residues, acetylation on lysine residues, and the opposing deacetylation. And then what I'm going to talk about today, which is protein methyltransferases or histone methyltransferases, which at specific lysine and arginine residues transfer a methyl group to these residues. And so if you look at the variety of enzymes which can covalently modify histones, you can see that the histone methyltransferases make about 50% of those enzymes. So here's a view of the human HMTome. So this is just a representation of the HMT family. So there's roughly 100 family members, and what you can see is that's roughly split down the middle, 50% being lysine methyltransferases, shown here, and the other half being arginine methyltransferases. I should point out that while lysine methyltransferases, there are known groups of lysine demethylases that we heard about from Jonathan Wettstein. There are no identified arginine methyltransferases to date. So those are the search for those is still ongoing if they exist. One other thing I want to point out on this family tree is that one of the enzymes I'm going to talk about today, which is DOT1L, is actually on the arginine methyltransferase tree. It actually conducts H3K79 monodi and trimethylation, but structurally, when you look at it phylogenetically, it actually looks more like an arginine methyltransferase. It lacks the canonical set domain, which all the other lysine methyltransferases have. The other highlight here is EZH2, which is a lysine methyltransferase, which I'm going to be talking about quite a bit today. So I want to give you a feel, at least at a high level, of sort of a genericized pathogenic mechanism that I'm going to be referring to today. And so what you have here is dysregulation of a particular HMT, histone methyltransferase, and that leads to site-specific hypermethylation, which is represented on this Western blot at a global level. And this hypermethylation can then lead to transcriptional changes, either aberrant upregulation or downregulation of gene transcription. And ultimately, this change in gene expression leads to phenotypic changes in the cell, and ultimately, this leads to disease. And for today's talk, we're of course referring to cancer. So then I'll give you the similar genericized therapeutic mechanism that I'll be referring to. So here we have a cancer with sensitivity or so-called a genetic addiction to a given histone methyltransferase. And we can see the site-specific methylation at a given histone residue. But if we intervene therapeutically with an HMT inhibitor, we can diminish that site-specific methylation and begin to reverse the aberrant gene transcription signature. And this ultimately leads to selective killing in genetically altered cells, but not in genetically normal cells. And ultimately, the hope is this contributes to tumor growth inhibition. So what sorts of genetic alterations confer dependence to HMT activity? There's quite a wide spectrum of them, and I've listed some of them here. So these alterations can be things such as point mutations, which I'll describe in detail for EZH2. It can be amplification or increased expression of certain HMTs. It can be loss of function of a corresponding demethylase, such as the demethylase UTX, which removes removes methylation from lysine 27 of histone 3 and opposes the EZH2 function. It can also be ectopic recruitment of a particular HMT to aberrant gene locations, and that's the story you'll hear today on DOT1L. It can involve indirect chromosomal translocations, such as the case with DOT1L, which I'll describe. They can also involve direct chromosomal translocations. So there's a very good example of this in myeloma. The HMT set undergoes a specific translocation with the immunoglobulin heavy chain promoter and seems to be a driver in a certain subset of multiple myeloma. And then finally, probably the harder to define is the so-called synthetic lethal relationships, where you have dysregulation in one pathway, such as switch-SNF, and that confers sensitivity to an HMT pathway, in this case, EZH2 or PRC2. So now I'll talk a bit about DOT1L and mixed lineage leukemia and some of our drug discovery efforts in that indication. So just to give you a little background on DOT1L, as I mentioned, it does not contain a set domain, but it is in fact a lysine methyltransferase. And it's believed to be the only KMT which actually methylates histone H3 K79, and it can do in vitro and seemingly so in cells mono, di, and trimethylation at that residue. As far as its normal role in terms of transcription, it seems to be involved in actively transcribed chromatin and has a potential role in transcriptional elongation. As you can see from this cartoon from a review from David Alice's lab, DOT1L has a number of different binding proteins such as AF10, AF17, and ENL, and AF9, and it can associate indirectly with the PTEF-B transcription elongation complex. So it seems to be involved with transcriptional elongation. But these binding proteins that I mentioned are very important, and I'll describe this in just a bit more detail in a moment, but essentially it's these binding proteins which cause DOT1L to either be directly or indirectly recruited by mixed lineage leukemia fusion proteins. And very importantly, work from Scott Armstrong and Jay Hess and others have shown that the DOT1L enzymatic activity is required for transformation in mixed lineage leukemia, both in the AML and ALL subtypes. So just a bit more about the actual MLL translocations. So MLL is in fact a histone methyltransferase. It's located at 11q23, chromosome 11q23, and what you see is here's the entire gene with a set domain at the very C-terminus, but what happens in the translocations is that this C-terminal portion of the gene is lost, and the fusion partner becomes a number of the proteins that I showed you in the previous slide. They're things that either bind DOT1L directly or things that bind indirectly to DOT1L. So this is the mechanism by which DOT1L gets recruited to MLL target genes. And as far as the incidence of the disease, MLL is rearranged in about 5% of ALL and about 5 to 10% of AML. But there's a very high proportion of infant acute leukemia which have MLL rearrangements. And I should point out that there's really a number of different fusion partners that have been described. There's over 80, and I'll show you some coverage as far as what we have to date on those fusion partners. So just a bit more about the mechanism and how DOT1L is recruited by MLL fusion proteins. Here we have the normal, quote-unquote, normal state. So if we're thinking about hemopoietic precursor cells, you have the MLL HMT at its target genes, and it's methylating histone H3K4, monodi and trimethylation. And you have a quote-unquote normal level of H3K79 methylation, which is very low, as you can see from this ChIP-seq plot. But what you, and so these hemopoietic precursors with this normal level of gene expression can undergo differentiation along the normal lineages. But what you have in the context of MLL is that the fusion protein actually recruits DOT1L by virtue of the fusion partner, and DOT1L is aberrantly methylating K79 monodi and tri, and that's what you see on this alignment here. And this aberrant methylation is leading to a prominent upregulation of the so-called MLL target genes. And these MLL target genes have been well defined by folks such as Rick Young, Scott Armstrong, and others. And two of the most prominent members of this signature are HOXA9 and MEIS1. Ultimately, the expression of this MLL signature leads to leukemogenesis. So just a few more points on the actual disease of mixed lineage leukemia in its current clinical setting. It has a very poor prognosis, both in the adult context and in the pediatric context. Less than 30% expected 5-year survival for both. These patients do get identified in the clinic currently. So the rearrangements are identified as part of normal cytogenetic workup and FISH for leukemia patients. But one important point is there really is unmet need for these patients. The current standard of care involves intense chemotherapy and stem cell transplantations. So to that point, we began a number of years ago, about 4 years ago, drug discovery efforts to find DOT1L inhibitors. And I'm really shamelessly sort of boiling down all the research and drug discovery for 4 years into this one slide. And through structure-based guided design, we took mimetics of S-adenosyl methionine, which is the substrate that HMTs use to transfer a methyl group to histones. And we did structure-based guided design to arrive at this aminonucleoside analog, which is EPZ5676, and it's our current clinical candidate. So this particular molecule has exquisite potency to DOT1L. It actually has a Ki that's in the double-digit picomolar range. It's also very, very selective. You can see that it's over 37,000-fold selective over other HMTs, and these little circles here are meant to represent potency on the HMT tree. The bigger the circle, the more potent the hit is on a given HMT. Very importantly, the effects that we have biochemically do translate into cellular activity, which isn't always the case that you see with some probe molecules. So you can see that we're getting a very nice inhibition of H3K79 methylation in this particular Western blot, the IC50 being around single-digit nanomolar. So as I mentioned, there's a key MLL target genes in mixed lineage leukemia. And we've been able to show in MLL rearranged cell lines when we treat with 5676 or other DOT1L inhibitors that we see a very nice time and dose-dependent inhibition of the target genes, HOXA9 and MEIS1. So really blunting the expression of these genes. You can note it does take some time, and that's consistent with the fact that you have to inhibit the enzyme, turn over the mark, and then have the downstream effects on gene expression. I should also point out that I didn't point out earlier, there is no known demethylase for H3K79. There hasn't been one identified, so it's thought that that turnover has to occur through actual turnover of histones in the cell cycle. So at a more global level, when we've done gene expression analysis, global transcriptional profiling, we see that the canonical mixed lineage leukemia signature is reversed when we treat with EPZ5676. This is a rearranged cell lines, this is MV411. We've also seen this in MOLM13s, which is another rearranged cell line. So the effects we have on methylmark and on transcription translate into anti-proliferative effects. And those anti-proliferative effects are specific to mixed lineage leukemia rearranged cell lines. So here in this slide, I'm showing you two cell lines. On the left, you see MV411s, which have an AF4 translocation. And on the right, you have a non-translocated cell line, just a Jurkat's cell line. And what you can see from the Western blotting at a global level, we're getting comparable amount of inhibition with increasing dose of H3K79. So we know the drug is getting on board, hitting the target, and inhibiting methylation. But you can see that only the rearranged cell lines really seem to care, and that's where you see the anti-proliferative effects. So it seems to be selective, our effects to MLL rearranged cell lines for the most part. And in fact, when we've looked more broadly across panels of MLL rearranged cell lines, we've been very successful in showing that, at least in a cell-based context, that we have proof of concept for anti-proliferative effects. And so I'm showing you here in these pie charts, just for infant, pediatric, and adult MLL, the frequency of the different translocation fusion partners. And the green is meant to represent where we have sensitivity, either using an inhibitor in a cell line or through work of our collaborator, Scott Armstrong, who's used a lot of engineered systems in mouse models to show the proof of concept for efficacy. So the cell line effects we can also translate into xenograft models, and what you're seeing here is a nude rat xenograft model. And you may ask yourself why you're doing nude rat xenograft models. Well, one thing I should point out is that one of the limitations of 5676 is it does have fairly poor oral bioavailability. It's very close to zero. So the drug actually has to be given by constant IV infusion, which you're seeing here. This is a 21-day constant IV infusion at 2 doses. You either have vehicle, 35 mg per kg per day, or 70 mg per kg per day. And what you can see is that if we cease dosing at 21 days and assess tumors, at both doses, we're getting nice anti-tumor effects. And in fact, at the higher dose, there are no more palpable tumors. If we then do a washout to see if the tumors come back, you can see at this highest dose that we've really wiped out the tumors. They don't grow back. Whereas this intermediate dose, you're seeing that we haven't quite hit the target hard enough, and the tumors begin to grow out during the washout period. Here at the bottom, you're seeing some of our effects on PD at the 2 doses. This is looking at within the tumor at H3K79 methylation, and this is actually looking in a surrogate tissue, the bone marrow, at H3K79 methylation. Similarly, the HOXA9 and MEIS1 gene expression, we also have nice PD effects there as well. So as I mentioned, EPZ5676 is a clinical candidate. It's currently in a phase 1 study, which initiated in September of last year. And we're currently in a dose escalation phase. And so we're currently taking patients with advanced hematological malignancies, including MLL rearranged patients. This is ongoing at 6 sites here in the US. We will be adding additional sites during our expansion phase. And we are looking for outcomes such as the maximum tolerated dose, early readouts on PK, and earlier effects on PD. So we're hoping later this year to move into our expansion cohort. We'll be stratifying for only mixed lineage leukemia rearranged patients, and we will be expanding to up to 12 sites in the US and Europe. And we'll be looking at outcome measures such as safety and getting an early assessment of therapeutic effect in MLL rearranged patients. So I want to share with you some data that we made public a few months back, which was very exciting for us to occur in our dose escalation phase. So we had a patient that enrolled in one of the early cohorts that was receiving a dose of 24 mg per kg, 24 mg per meter squared per day, which we believe is really below the therapeutic dose, which we estimate potentially to be somewhere between 54 to 80 mg per meter squared per day. Depending on what cell model we base that on. But what we saw in this patient who had a rearrangement in ALL was that we saw a 60% reduction in the methyl mark in the PBMCs. But what was even more striking is that we saw a biological effect. The patient had resolution of fever by day 5 and had a marked decrease in blast count. Unfortunately, this patient had very late-stage ALL and began to suffer disease progression into the CNS, so had to come off study. But this was at least a promising result for the drug in this early phase of the trial. So stay tuned as we move into the expansion phase for more results to come. So I mentioned that we really only see effects, antiproliferative effects in MLL rearranged cell lines. And that's been true with one exception to date, and that is that we have found that a certain type of AML that contains a genetic alteration called MLL partial tandem duplication, or PTD, seems to be responsive to DOT1L inhibitors. So for those of you who are not familiar with MLL-PTD, what it actually is an internal tandem duplication within the MLL gene where exons 3 through 9 are actually internally duplicated. Now, what's puzzling is there's really no obvious reason as to why this would confer sensitivity to DOT1L. Yes, there's a connection to MLL, but there's no translocation bringing a fusion partner that binds DOT1L on board. So we're still, this is still an area of active investigation for us and for our collaborator Scott Armstrong as to why there could be sensitivity in MLL-PTD. There's really only one MLL-PTD cell line that we've been able to obtain today and test, which you see here. We're getting comparable sensitivity in the low nanomolar range as we see with the MLL rearranged cell lines. And we also are able to show that we see efficacy in xenograft models with this cell line. We're hoping to bring more MLL-PTD cell lines on board. There's really only a couple that we've identified in the world. And so we're hoping to really pursue this as an area for clinical development. So I'll transition now from DOT1L to tell you more about another story which involves EZH2 and non-Hodgkin lymphoma. So I could spend hours going over background EZH2 and its connection with cancer, but I really want to get to data, so I'm just going to give you a brief background on EZH2. So it is the catalytic subunit of the so-called polycomb repressive complex 2, which is shown here. It's a multi-protein complex, generally about 4 members, which EZH2 is the catalytic subunit. And it generally targets and represses genes involved in development and cell fate. EZH2 is a set domain containing methyltransferase, and it generates mono, di, and trimethylation at histone H3 lysine 27. And there's been compelling evidence in the literature that EZH2 has a role in cancer. It's been shown in a number of different contexts to be genetically altered in cancer. It's been shown to be an oncogene both in vitro and in vivo, with the catalytic activity being required. And it's been shown through a number of knockdown studies and now small molecule studies to be involved in tumor cell growth both in vitro and in vivo. So going back a few years, a group at the British Columbia Cancer Agency put out a paper where they were doing sequencing in non-Hodgkin lymphoma, and they reported some point mutations in EZH2 that occurred in about 7% of follicular lymphoma and about 31% of diffuse large B-cell lymphoma, and this was exclusively in the germinal B subtype, not in the ABC subtype. Also wasn't seen in MCL or any other types of non-Hodgkin lymphoma. And so more recent data has suggested that the frequency of the mutation is probably more like 20% in diffuse large B, germinal center B subtype, and follicular lymphoma. But the mutation in question was recurrent, and it occurred at a specific tyrosine residue in the active site, tyrosine 641. You'll also see this in the literature referred to as tyrosine 646. It just depends on which reference transcript you're using. But this group reported that the tyrosine 641 could be mutated in patients to phenylalanine, asparagine, histidine, serine, or cysteine. But in all cases, the mutation was heterozygous. Only one copy was mutated. And again, it was occurring at a single amino acid. When this paper came out, the group tried incubating recombinantly expressed forms of these mutant enzymes in the PRC2 complex with naked peptide, and by naked I mean unmethylated versions of H3K27. And what they saw compared to wild type is that there really was a blunting of enzymatic activity. So they reported these were loss of function mutations in EZH2. We were somewhat skeptical about the finding, primarily because of the compelling literature on EZH2 in cancer, the fact that the mutation was heterozygous, and the fact it was occurring at a single amino acid. So we thought there should more investigation should go forward. And so what we did was take the recombinant mutant enzymes in the PRC2 complex and incubate them with increasing methylated forms of the K27 peptide. So in blue is unmethylated peptide, red is monomethylated K27 peptide, and yellow is dimethylated. And what you see for wild type is that it really prefers unmethylated K27 peptide. It's really good at generating dimethyl K27 from monomethyl, less good at taking mono and making di, and even worse at taking dimethyl and making tri. And what you see when you look across the mutant enzymes is exactly the opposite profile. They're not very good at taking the unmethylated peptide and making dimethyl, which is why the authors showed that there really was no activity on unmethylated peptide. But when you start looking at monomethyl, you see increasing activity, and then when you use dimethyl peptide, they're very good at making trimethyl, H3K27 from dimethyl. So this occurred to us that these were probably change of function mutations rather than loss of function mutations. So if you look at the actual enzyme kinetics, it really explains why a heterozygous EZH2 mutation at this particular amino acid is sort of the perfect storm for giving you hypertrimethylation. So if you take just wild type EZH2 and you add naked peptide, you get only a very modest or mild production of H3K27 because this particular enzyme is not very good at taking the higher methylated forms and making trimethyl H3K27. When you look at the mutant enzyme alone, it's not very good at taking the unmethylated and generating the higher forms the more methylated forms of H3K27. So you get a really puny amount of H3K27 trimethylation. But when you have a mixture, a heterozygous mixture of the wild type enzyme and one of the mutant enzymes, the wild type enzyme can generate the monomethyl and the di, while the mutant enzyme is really good at generating the tri from the di. And so you get a robust H3K27 trimethylation, and that's represented here. And in fact, when you look in cell models, which essentially are derived from patients and have been sequenced and shown to have Y641 mutations, they're shown here on the right side of the Western blot, what you see is that these cell lines, which are diffuse large B-cell lymphoma, germinal center B subtype, you see that they have hypertrimethylation of H3K27 and very little dimethylation, and you see just the inverse when you look at the wild-type cell lines. So in thinking about a drug discovery effort for EZH2, we began several years back again to do high-throughput screening towards the PRC2 complex. And doing our HTS and following that with iterative SAR, we took our initial hit and we drove it to a very potent selective inhibitor of EZH2. And this compound, EPZ6438, also referred to as E7438 to indicate our partner Eisai's nomenclature. This is a SAM-competitive, very potent and selective inhibitor of EZH2. So it's about, it has a single-digit nanomolar Ki towards the enzyme, and it's 4,500-fold selective over 14 other HMTs that we've looked at, and 35-fold selective over the closely related EZH1. So it's both potent and selective for EZH2. And this mechanistic data, I'd be remiss, and Bob would be mad if I didn't mention, this is a SAM-competitive inhibitor, and it's non-competitive with nucleosome. So when we look back in those cell lines that I showed you just a moment ago with hypertrimethylation of H3K27, what we see is we get very potent and selective cell killing. So here we're looking in a Y646F mutant form of the enzyme in this WSU cell line, which again is a GCB subtype of diffuse large B-cell lymphoma. And we can see that we're getting very robust anti-proliferative effects and nice effects on methyl mark, about single-digit nanomolar. In this non-mutant form of EZH2 in this OCL cell line, which is also a GCB subtype, we're getting comparable inhibition of the methyl mark, but you can see the cells, in these cells, there's very little anti-proliferative effects. So the anti-proliferative effects seem to be specific to the mutant cell types. And that's represented more so on this graph. Here on the y-axis, what I'm showing is a metric we use called lowest cytotoxic concentration. So it's a mathematical treatment of anti-proliferative data, and what it really means is that it's the dose at which you induce cell stasis in an 11-day growth assay. So if you're above the LCC, cells are progressing, continuing to grow, and if you're below the LCC, you're driving cell death. And so what you see is that as the mutant GCB cell lines tend to be an order of magnitude, and oftentimes 2 orders of magnitude, more sensitive than non-mutant cell lines. There are 2 outliers to that statement, here and here. One of them is this RL cell line, which is a 646N mutation, and both our group as well as groups at GSK and at other institutes have reported that this cell line is resistant to EZH2 inhibitors for reasons we don't yet understand. The other outlier is this exquisitely sensitive cell line, which is actually a follicular lymphoma, and it's this so-called Pfeiffer cell line, which has a different type of mutation, A682G. I'm going to come back to that in just a moment, but it seems to be exquisitely sensitive to the EZH2 inhibitor. So we're able to translate the effects that we see in cell culture into nice efficacy in xenograft models. So here we're looking at a model of a Y646N mutant diffuse large B-cell lymphoma. This is a Carpus cell line, Carpus 422, where we're dosing with increasing doses of EZH2 inhibitor, 6438, BID orally, 80 mg per kg, 161 mg per kg, and 322 mg per kg per day. And so this drug was very well tolerated in the animals, no obvious effects on body weight or any signs. And what you can see is that you're getting increasing amounts of tumor growth inhibition. And in fact, at these 2 higher doses, you've really wiped out the tumors. In fact, if we do a washout period, as I showed you in that DOT1L xenograft study, we can see that these tumors remain completely wiped out for an additional 30 days or so in this study. Accompanying the efficacy data you see on the right here, very nice PD data. We're looking at trimethylation of H3K27 in the tumor sample using an ELISA assay, and you can see that we're getting nice dose-dependent effects on methylation in the tumor. These are just some other surrogate tissues to show you that we can detect H3K27 methylation modulation in tissues outside of the tumor. We can see it in the PBMCs, we can see in bone marrow, and we can see in certain skin cell subtypes. It's not to say that all tissues that you see an effect on H3K27, but we certainly have identified some where you could detect that change. So I want to return to the story I mentioned just a moment ago with the Pfeiffer mutation, the A682G. So there were some additional publication. There was an additional publication that came out from the BCCA group a few years back, as well as a publication from Todd Golub's lab, which identified non-Y641 mutations in non-Hodgkin lymphoma. And there were really just one incident of the A682G mutation and one incident of this, one sample of this A682V mutation in this study, and only one of the 692V mutation in the Golub paper. So it's thought that these mutations were quite rare, and they are lower frequency than the 646 mutations. But some recent work from Jude Fitzgibbons was published recently in Blood. It's actually presented at ASH last year. But this group identified in a sampling or sequencing of 366 cases that 9 of them were A682G and about 7 were the 692V mutation. And in both cases, this was in follicular lymphoma rather than the diffuse large B-cell lymphoma. And so our group, as well as other groups, have characterized these mutations as we did with the 646 mutation. And again, we see that these are change of function mutations. The A682G mutation seems to be particularly robust in using all forms of K27 peptide. So it seems to be a very active form of EZH2. So we believe that, again, that these Pfeiffer cells represent a particularly hypersensitive subpopulation of diffuse large B-cell lymphoma. Here we're presenting you with a Pfeiffer xenograft study where we're doing just once-a-day dosing with EPZ 6438 in this Pfeiffer cell model xenografted in nude mice. And what you see is that at increasing doses of compound, you've really wiped out the tumors at the highest 3 doses. And at this lower dose, you cause a tumor stasis. But if we wash out, we can see the higher doses, you've really got durable regressions, whereas in this lowest dose, you haven't quite hit the tumor hard enough, and the tumors come back during the washout period. So EPZ6438 is our clinical candidate, and once again, we've initiated clinical trials with this molecule. This started in June of this year. The trial is currently in a dose escalation phase. It's for patients with advanced solid or hematological malignancies, including EZH2 mutated non-Hodgkin lymphoma. We currently have 2 sites active. One is the IGR in Paris, and another is the Institut Burgundy, and also in France. And we're currently looking for our maximum tolerated dose, and we're getting reads on PK and PD. We're hoping in 2014 to move into our expansion phase, where we'll start restricting to patients with relapsed or refractory EZH2 mutated non-Hodgkin lymphoma. And we'll, of course, be expanding our number of sites on a multinational basis, and we'll be looking at measures such as safety and getting an early read on potential therapeutic benefit. So the final story I want to leave you with has to do with additional indications for EZH2, and this is an exciting story in a particular cancer type where there really is unmet need. So I mentioned earlier that one of the prominent ways in which chromatin is regulated is through covalent modifications on histones. But there's another mechanism that involves the so-called chromatin remodeling complexes. And one of the most well-known members of this chromatin remodeling complexes is the so-called switch-SNF complex, which is shown here. The switch-SNF complex is conserved among eukaryotes. It's found all the way down to yeast, but it's a very, very big complex that involves up to 14 different proteins. And the composition of this complex in terms of what proteins are in the complex can vary quite a bit. There's at least 28 genes encoding these proteins. But this is an ATP-dependent complex, and what it essentially does is it's involved in histone positioning. It actually can affect the occupancy of histones at promoters, but it's been shown by a number of different groups to have a tumor suppressor function. I think it's probably a little bit of an oversimplification to call it a tumor suppressor, but in a number of different contexts, when you have loss of function of members of this complex, there are oncogenic effects that are observed. So one such case was reported by Charlie Roberts and Stu Orkin a couple of years ago, and that's what got us interested in this story. This group was looking at the role of switch-SNF in its relationship with PRC2, the complex which has EZH2. And what they report in their model is that in a normal circumstance, switch-SNF seems to have somewhat of an antagonistic relationship with PRC2. That's both at the level of transcription of EZH2 and at the level of the actual polycomb target genes. And so this antagonism is important for keeping stem cell programs suppressed. So in the case of a particular type of tumors called rhabdoid tumors, which is a pediatric cancer, which I'll mention in just a moment, greater than 90% of these tumors have a biallelic loss of function of a member of the switch-SNF complex called SNF5. I should point out that SNF5 has the honor of having more names than you can probably keep up with. It's called SNF5, it's called BAF47, SMARCB1, and INI1, but it's all referring to the same protein. So I'll try to refer back to those different names. So SMARCB1, as I mentioned, is the same thing as SNF5. But beyond rhabdoid tumor, there's been a recent report, as a review from, I believe, from Jerry Crabtree's lab that showed that probably around 20% of all human cancers have some loss of function in members of the switch-SNF family. Oh, I should just back up one moment. So in the case of the rhabdoid tumor, what you have is the loss of function of SNF5 that alleviates the antagonism that switch-SNF is having on PRC2 and on the target genes. This alleviates the inhibition on stem cell programs, causes potentiation, and ultimately contributes to oncogenic transformation. So just a few more points on rhabdoid tumors. So SNF5, as I mentioned, is mutated in about 98% of malignant rhabdoid tumors and in atypical tetroid rhabdoid tumor. These are very aggressive tumors. They're pediatric, and they're cancers of the brain, kidney, and soft tissues. And currently, there really is no effective treatment. It's very sad. The patients generally die within 1 year of diagnosis. But another feature of these tumors that's quite interesting is that the SNF5 loss of function is really the sole recurrent genetic alteration, which really suggests that this particular chromatin modulating complex may be a loss of function that is a driver in the disease. So we decided to test an EZH2 inhibitor in model cell lines for malignant rhabdoid tumor. And that's what you see here. This G401 cell line lacks expression of SNF5, has biallelic loss of SNF5. And what you see is a fairly robust cell killing in these cell lines compared to vehicle. You can also contrast that to what we see in a control cell line. And I use the term control cell line somewhat loosely here. This is actually a rhabdomyosarcoma, which isn't really a perfect control. It has nothing to do with rhabdoid tumor, but there's really no examples of rhabdoid tumor that, where we can get a hold of where there isn't a biallelic loss of SNF5. So this will have to suffice. But you see there's no effects of the EZH2 inhibitor in this cell line. When we've looked across other cell lines that don't have loss of function of SNF5, we've also seen that they don't respond to the inhibitor. I'm just showing some of those here, some 293Ts and another cell line which have expression of SNF5. When we've looked in other SNF5 mutant cell lines which don't have any expression, we see that they're generally about an order of magnitude or two more sensitive to the inhibitor. So as far as what's going on at a more of a mechanistic level, what we see is that in these MRT cell lines with SNF5 loss of function, we're inducing a G1 arrest. It takes a while. It takes about 7 days to start to see an arrest in G1. And just following that, you start to see an increase in sub-G1 population and tunnel positive cells about around at day 11 and even more so at day 14. If you contrast that to what you see in that RD SNF5 wild type cell line, you can see there's very few effects at all on the cell cycle profile. So again, it's very specific to the SNF5 mutant context. When we've done global transcriptional analysis on SNF5 deleted cells, it's quite profound that the difference at a global level, what you're seeing in terms of transcriptional effects. So just looking at a very global level, starting around day 4, you see a very robust increase in gene expression consistent with alleviation of PRC2 enzymatic activity, and that's even more robust by day 7. But you can contrast that with what we see at a global level in the SNF5 wild-type cell line. Very few changes in gene expression, even out to day 7, only just over 100 or so. So we've been looking more closely at what pathways are being affected in these cells. What we're observing is that we're normalizing the canonical dysregulated pathways in MRT. So you see here the hedgehog pathway, which is typically upregulated in MRT, and we're causing downregulation of genes such as GLI1 and PATCH1 in a time-dependent manner. We're also upregulating markers of neuronal differentiation, which are typically downregulated in MRT. So we see DOC4 up, CD133 up, and PTPRK up, but only in the G401 cell line. We also see upregulation of tumor suppressors such as BIN1, cell cycle inhibitors such as P16 and P21, again only in the mutant context, and then downregulation of the MYC oncogene. So we've been able to show that in xenograft models using the G401 cell line that we do have nice anti-tumor growth inhibition effects in a dose-dependent manner here. Again, dosing BID with 6438, increasing concentrations of the compound. Again, we're dosing for 28 days, doing a washout, and looking for durable regression. And you can see at the 2 higher doses, the tumors are completely wiped out and don't come back during the washout period, whereas the lowest dose, you can see that we haven't hit the target hard enough. Again, the drug is very well tolerated, no real effects on body weight. We see nice PD effects, as you see in this immunohistochemistry staining. We're really blunting H3K27 trimethylation in the tumor and the dosed animals. And we're also seeing similar effects on gene expression, on the neuronal differentiation markers, and on the hedgehog pathway, as we saw in the cell lines. So I just wanted to leave you with one additional thought about so-called INI1, which is SNF5 deficient tumors. So there's really a spectrum of INI1 deficient tumors, particularly in soft tissue sarcomas. And shown here on this graph is just a flavor of some of these very rare tumor types. They consist of things such as epithelioid sarcoma, synovial sarcoma, atypical chordoma, and extraskeletal myxoid chondrosarcoma. In each of these, there's a varying degree of loss of INI1 or SNF5. And so we're hopeful that they may represent additional indications for EZH2 inhibitors. And to that point, I just wanted to highlight the synovial sarcoma for a moment. So in synovial sarcoma, there's a hallmark translocation where a gene called SS18, which is located on chromosome 18, is translocated to one of a few genes in the SSX family, which is located on chromosome X, and you get this fusion product. And this is important because SS18 is normally found within the BAF or switch-SNF complex. It's actually one of the proteins that comprises this complex, and it's bound here to BAF47, which again, SNF5, INI1, SMARCB1. And so what you see in the translocated context is that the fusion protein actually binds to the switch-SNF complex and displaces the wild type SS18 and BAF47 or SNF5. And this gets proteolytically degraded and causes a SNF5 deficiency. This is from a review from Jerry Crabtree's lab, by the way. I'm not showing the data today, but we've been able to obtain some synovial sarcoma cell lines, and we're seeing very nice anti-proliferative effects in those cell lines, and we're hoping to continue to expand those studies and investigate further in the clinic synovial sarcoma as an indication for EZH2 inhibitors. So we're very hopeful in that area. So I hope that I've given you a feel for what Epizyme's philosophy is on drug discovery. We're starting with genetically defined cancer indications where there is sensitivity to a given HMT, doing our drug discoveries for that HMT, and then using cell models and xenograft models for those genetically defined cancers, and then translating that hopefully into clinical efficacy as well. And so with that, I want to thank all the folks who were involved in this with this data, and there's quite a few of them shown here. The folks at Epizyme in discovery are shown in this column, and development and clinical at Epizyme here. These are academic collaborators, Scott Armstrong and Catherine Berndt. And then finally, our colleagues at ASI who are involved in the clinical development of the EZH2 inhibitor. So I'll close with that and take any questions.
One burning question, then I'll
Sure.
turn it over to the other guys. So myelodysplastic syndrome, Malignancy, has reasonably high frequency of EZH2 mutations that really look to be loss of function, focal microdeletions or biallelic inactivation. So first of all, that's interesting.
Yeah.
These are clearly gain of function. Those are loss of function. Two different heme malignancies. And the other, so it'd be interesting to hear what you think about that. But also, does it in some way represent a caveat for sustained inhibition of EZH2?
Yeah, it's a good point. So it's very true that EZH2 is not just strictly an oncogene. There are cases where it seems to be more of a tumor suppressor. There's also the work from David Alice's lab on the histone mutations and inhibition of EZH2 in glioblastoma. What I would say to that is two things. One is that very often those mutations are in the context of other mutations, other drivers, and so it's unclear that EZH2 is the sole genetic mutation is the sole genetic event in MDS. The other thing I would point out is that we're hoping that in our clinical trials to really be in acute dosing and not in chronic dosing, but we'll just have to wait and see. But in general, we just have to be aware of those caveats and really keep our eye out for anything that pops up related to that.
Thanks.
Yeah.
So Jesse, the washout experiment where the tumor's gone and then you looked at methylation, did you guys go back and monitor various tissues for their methylation return? And is one, and number two is when you did look across the tissues, were there any tissues that seemed refractory to the drug?
To it coming back. There are definitely tissues where you do not see inhibition of methylation. For example, if you look within the skin, in the hair follicle, you see no inhibition of H3K27 methylation, but in a layer of the epidermis, you do see it. So some of this can be explained by two things. One is that EZH1 it does have an expression pattern that's different from EZH2. They're not completely redundant. So in some tissues, there might be some redundancy there. It's also true that just basically different tissues are going to have different amounts of drug on board. We have looked at different tissues to see, what the bounce back is for the methylation. You can detect it come back, say, in PBMCs, and it doesn't come back at the same rate in all tissues, and certainly not all tissues have the same sensitivity. Yeah.
So, excellent presentation. I have a question regarding your shortcut experiment. I'm sure many people ask the same question.
For what experiment?
The shortcut experiment.
Oh, shortcut, sorry, yes.
The DOTIL inhibitor inhibits both the rearrange and the shortcut methylation at the same level. So can you comment whether there is the same gene transcription effects and why shortcuts are refractory to this drug while they
Right.
targeting this substrate?
Right. So Scott Armstrong's really done a lot of work in understanding what DOT1L is doing in a non-rearranged context. And I think the sort of short answer is it's really only a fine tuner in the non-rearranged context. It may fine-tune elongation, but doesn't seem to be a master regulator. I think that's the way I would describe it. So that only in the rearranged context where you're recruiting it where it's not supposed to be and starting to get aberrant methylation are you conferring sensitivity. You just don't, when you look at the genes that are affected by a DOT1L inhibitor in a non-rearranged cell line, there's just a very few genes that are affected. It's only a very small handful, and when you do the ChIP-seq with that, Armstrong's published on this, there's just not a lot of effects on those genes.
And those genes have anything to do with survival effects?
Not really. I mean, like actin is one of the ones that loses methylation and probably doesn't have an effect on gene expression. There are some where there's both an effect on the methylation and gene expression, but there's nothing you can really say about them as to, that they're pro-survival or have any sort of canonical pathway. Yeah.
So,
Bruce.
Is the mechanism of tumor regression, is it differentiation or apoptosis?
That's a great question. So that's something we're really studying a lot right now. You're talking about for DOT1L, I assume?
Yes.
Yeah. So the answer is yes, it's both. So what we see is that it really seems to be a differentiation effect where there's it's very temporarily, not very temporally, excuse me, let me rephrase it. The timing of onset of apoptosis seems to be very close to the onset of differentiation. At least that's in tissue culture. Now, that may not be the same in patients. We just don't know yet. But what we're at least seeing in tissue culture dishes is that you're inducing markers of differentiation. You're inducing phenotype of differentiation by microscopy. But very soon thereafter, you're starting to induce apoptosis. It's hard to temporarily disconnect them.
Lou.
So there's all sorts of deletions, mutations of various chromatin modifying components in cancer. Is it the case that it's just this particular SWI/SNF complex that is antagonizing PRC2 or others?
Yeah, so with regard to the switch-NIF complex, let me say one thing. First thing is that we don't think that all situations where you have some loss of function or change of functions in switch-NIF are you going to be sensitive to EZH2, and that's
the
definitely true. And is that the question you're asking?
Exactly.
Yeah, no, that's fine. So in some of the epithelial cancers that we've looked at, like lung, they don't seem to be as sensitive to an EZH2 inhibitor, and maybe because there's just so many other genetic aberrations in those cancers that it's just not as clean.
I have one other quick kind of technical question. So for the DOT1 trials, as you said, the MLL rearrangements were good at detecting those with routine genetics, but the PTDs are cryptic.
Yeah, they are.
And a lot of clinical labs struggle with PCR-based or Sanger-based. So how are you going to handle that?
It's a great question. I mean, we've really been talking to a lot of advisors, folks and clinicians about how to pursue this. And so we're looking into that right now. I don't think I have a definitive answer for you, but it probably is going to have to be something that's PCR-based. But yeah, it's something that we're really looking into because there's just no easy solution there.